Dear students, Namaskar. I'm Dr. Nutan Prakash Vishwakarma, and in this particular video, I'm going to talk about some methodologies for the phylogenetic analysis. In my previous videos, in the lecture 1 and lecture 2, I've discussed a lot about what phylogenetic analysis is all about, what are the basic concepts in that, and in this video, we'll try to figure it out that how many methods are available for the phylogenetic analysis. So once you decide to go for phylogenetic analysis, the main question comes in your mind that which particular sequence you should target for the evolutionary study among the organism or in a particular gene or in particular proteins. The best way to go for the phylogenetic analysis of the organism as a whole, you should have the whole genome sequence. Say for example, you want to study the phylogenetic relationship among 50 of organisms. So the primary requirement, first of all, you should have the whole genome of all the 50 organisms, which is currently not possible in our public domain databases. There is unavailability of all the organisms genome sequence. What should we do? Until the whole genome sequence of all the organisms are not available, how we can go for phylogenetic analysis? For that one, we need some representative sequence which size is less, and although whole genome sequence is not available, that particular sequence will act as a representative sequence of that particular organism. So that is known as molecular chronometers. So why we use molecular chronometers? Rather than taking the whole genome sequence of organism, we target some representative sequence, and based on that we can go for phylogenetic analysis. So the concept of the molecular chronometer was first of all given by Zuckerland and Pauling in 1965. So maybe the molecular chronometer may be highly conjugate protein, just like example is ubiquit, or maybe it is nucleic acid like rRNA, whose rate of mutation is constant. And as time passes, bacteria evolve and the sequence of the molecular chronometer slowly changes. Thus, organisms with fewer changes are more closely related. So, what should be the criteria to select the any particular sequence as a molecular chronometer? Number one, whatever sequence you are targeting, whatever sequence you are taking, that should be present in all the representative of the group study. So, for example, if you want to study the phylogenetic relationship among the organism, so whatever sequence you are taking, that should be present in all the organisms of that particular domain. So for example, you are well aware about the 16S rDNA sequence. So 16S rDNA sequence is present everywhere in the prokaryotic domain. So 16S rDNA represents as a molecule, can be taken as a molecular chronometer. The second condition to be qualified for the molecular chronometer is function must be same in all the representatives, means the function of that particular sequence should be conserved. Irrespective in any organism, the function should be same. Point number 3, sufficient level of similarity should be there in the sequence of all the organism. And definitely, some level of differences should be there. So based on that, we can study the phylogenetic relationship. So here I am giving you some example of molecular chronometers. Say for example, if you are studying the phylogenetic relationship among the organism in the animals, you can take hemoglobin as a molecular chronometer. The function of hemoglobin in all the organisms are same, like that is oxygen carrier, and it is present in all the animals. If you want to study the phylogenetic relationship among the aerobes, you can take the cytochrome C, that is actually protein, and it is the function is in respiration. ATPase can be taken as molecular chronometers. The function of it is in ATP synthesis, and it is present in animal, plant, and microbes everywhere. Rec-A protein, it takes part in the homologous recombination, and it is present in almost all the organisms. 16S rRNA, nucleic acid, obviously, it's taking part in the ribosomal structure, and it's present in all the cellular life. So there are 3 rRNA molecules in prokaryote, namely 5S rRNA, 16S, and 23S. So for the microbial identification purpose, or for the microbial diversity study, or for for the microbial phylogenetic analysis, we take 16S rRNA for the study. However, in the eukaryotic phylogenetic analysis, we use 18S rRNA gene, means 18S RDNA. Either you say 18S rRNA gene, or you say 18S RDNA. All right. Either you say 16S rRNA gene, or you also can say 16S RDNA gene. So once you have chosen the molecular chronometer and you want to study the phylogenetic relationship among the organisms, then what are the different criteria, what are the different methods available? With the help of that, you can do the phylogenetic analysis. So there is mainly two methods available. The number one is the character based, and number two is the distance based. What happens actually in the character based? Say for example, here I have 5 species A, B, C, D, and E. So in the character based method, what we do, first we go for the multiple sequence alignment among the sequences. So whenever you have to perform the phylogenetic analysis, the first method should be the multiple sequence alignment. So once you perform the multiple sequence alignment, in the aligned characters, like it may be DNA or it may be protein sequence, directly we conclude about the structure. We conclude about the phylogenetic relationship among the species or organism. How we do it, we'll see in couple of minutes, that what character based method is actually all about. The second method is means directly we focus on the characters, and the second method is known as distance based method. In the distance based method, what we do actually, we first perform the multiple sequence alignment, and then we generate a kind of the matrix in which the distances between the sequences we put. Say for example, let me take an analogy. Suppose a professor is taking your lecture. So the student who is sitting in the first row of the class, he have the minimum distance with the professor, and the last bencher have the maximum distance with the professor. So if I give a matrix in which in respect of the professor, there is the distances have been given, that student A is 1 meter from the professor, student B is 20 meters from the professor. So by just seeing that value, we will come to know that which one is more closer to, which student is closer to professor. Likewise, once we do the multiple sequence alignment, then on the basis of the similarity, we calculate the distance matrix. How we actually calculate the distance matrix, we'll see it once we'll deal with the distance based method. So based on that, we generate the distance based this kind of the matrix, and based on this particular values, we make the phylogenetic tree. We'll see later on once we'll deal with the distance based method. So right now we are going to discuss about the character based method. Here is the summary. You can see there is two type of methods available for the phylogenetic tree building methods. The number one is the character based, number two is the distance based. Character based, there is two method, the maximum parsimony and number two is maximum likelihood. In the distance based method, there is two theory. Number one is the minimum evolution and number two is the least square. So with the help of the minimum evolution or least square, you generate actually the distance matrix, And based on that distance matrix, there is two more algorithm comes. One is known as UPGMA and second is known as never joining methods. So with the help of the UPGMA, we can make the cluster and we can generate the tree. Likewise, we also can use the never joining method to construct the tree. What are these all about? We'll see one by one. So now we are focusing on the maximum parsimony. Then you will come to know slowly. You will come to know what are the different methods and what are the logics behind it. So now I'm going to tell you what maximum parsimony is. So the most parsimonious tree is the one that requires the fewest number of evolutionary events, like the nucleotide substitution, amino acid replacement, to explain the sequence. If you see that what parsimony is all about in the dictionary, you will come to know that is the most economic way to represent the phylogenetic relationship is known as parsimony method. Let me give you an example. You will come to know. Say for example, you have to travel from one city to another city. So what are the most parsimonious way you can travel from city A to city B? So you have the different options available with you. You can drive your own car. You can go by train. You can go by flight. All right. So the method, the transportation way in which maximum means minimum money you have to spend, that will be the most maximum parsimony method. Means if you go by train, that will be most cheaper. Likewise, once you want to establish the evolutionary relationship among the organism or taxa or species, you can say the tree in which the minimum number of mutation takes place, that is going to be called as maximum parsimonious tree. So the advantage of maximum parsimony method is it is very simple and logical. Even with the help of pencil and paper, you can draw the conclusion. The next advantage is the maximum parsimony method can be used on molecular and non-molecular data also. It can be used for character, means it can infer the exact substitution analysis. Number 4, it can be used to infer the sequence of the extinct, means hypothetical ancestor. The only disadvantage of using the maximum parsimony is it is simple, intuitive, and logical, but it is not supported statistically. All right. So let's see in very detail. So the concept of parsimony is at the heart of all character-based method of phylogenetic tree reconstruction. So the two fundamental idea of biological parsimony are that mutations are exceedingly rare events. Mutations are very rare. Number 2, the more unlikely event a model evokes, the less likely the model is to be correct. So as a result, the relationship that requires the fewest number of mutations to explain the current state of the sequence being considered is the relationship what is most likely to be correct. So now let's try to understand this maximum parsimony method with the help of the example. Okay. So here, let me directly show you. Okay. Let me directly show you the sequence so that you can understand how we calculate the maximum parsimony tree. So for example, I have 4 sequences and I want to know that suppose sequence 1, 2, 3 and 4, that what is the relationship between sequence 1, 2, 3 and 4? What are the biological relationship? What are the evolutionary relations among them? I want to use the maximum parsimony method in that. So as I already I already told you that whenever you want to study the phylogenetic relationship, the first step you should go for multiple sequence alignment. All right. So suppose here I have performed the multiple sequence alignment. Now in these multiply aligned sequences, what first of all I will do to reduce the time and calculation, I'm not going to include all the bases. Only few of the bases I'm going to include to conclude my result. So the basis which I'm going to include in my result, that is going to in my analysis, that is going to be called as an informative site, and whichever is not going to be selected, that is going to be called as a non-informative site. So let's understand how you will decide that which one is the non-informative and which one is the informative site. So now I am telling you one by one the condition to qualify as a informative site. So the number one, the condition number one that you see in a particular column. So here suppose I am observing the column number one. So you concentrate yourself on this particular column. To be called as an informative site, there should be at least two type of the bases present in the column. At least two type. If it is more than that, there is no problem. So here you can see that in the column number one, only one type of the base is there. So only G is there. So obviously it is not informative site. Now the condition number two. The condition number two says that both the bases should be repeated twice, at least twice. Let me repeat the concept. Number one, to be called as an informative site, in any particular column, at least two type of the bases should be there. And point number two, both the bases should be repeated at least twice. If they repeat more than that, that's not a problem. So see in the column number two, G is repeating thrice, but A don't have any repetition, means both the bases don't have the repeated time. Twicely they are not repeating, so 2 is again not qualifying the being a informative site. What about the column number 3? In column number 3, here I can see that uh, G is repeating, there is at least two type of the base should be there. So here you can see there is three type of the bases. So condition number 1 is qualified, but at least two of them should be repeated twice. Okay, so G is repeating twice, but A don't have any repetition, T also don't have repetition, so 3 also doesn't qualify as a informative site. What about the 4? In 4 again you can see four type of bases are there and none of them is repeating twice. So again 4 is not informative site. What about the 5? Column number 5, you can see the two type of base are there, G and A, and both of them are repeating twice. So this is the informative site. In the column number 6, again you can see two type of bases there, G and T, and both are repeating twice. So here you can see that column number 5 and column number 6 are informative. So in this way, you can choose the informative side, and in uninformative side, you have to concentrate on only informative sites and we can leave the uninformative sites, means in the uninformative sites we are not going to include in the our calculation. Let me take more examples so that you can understand everything. One more example I have given here. Suppose you have the sequence 1, 2, 3 and 4. You multiply align the sequences. Now you have to perform the phylogenetic analysis by the help of the maximum parsimony method. So first of all, you have to choose the informative site. So is column number 1 is the informative site? No, obviously. Why? There is only a single type of basis present. To be selected as an informative site, at least two type of base should be there. So obviously column number 1 is not informative site. What about the 2? In the column number 2, there is two type of the bases, A and G, but G is repeating thrice, but A don't have any repetition. So 2 is again not informative site. So we can leave the 2 also. In the 3, there is three type of bases, but only A is repeating twice. Either C or G, both don't have any repetition. So 3 is also not informative site. What about the 4? Four type of bases are there and none of them are repeating twice. So again, column number 4 is also not informative site. What about the 5th? In the fifth, you can see two type of bases there, and both are repeating twice. So this column number 5 is going to be called as the informative site, and for ease, for simplify, so that we can identify, here I have put it the star, means if this is informative site. In the column number 6, you can see only single type of basis present, that is T. So this is not an informative site. In the column number 7, G is repeating twice, C is repeating twice. So column number 7 is informative site. Column number 8 is also not informative site because only one type of base is present. In the column number 1, column number 9, you can see that 2 times A is present and 2 times G is present. So now I conclude that column number 5, 7 and 9 all are informative sites. So I'm extracting the column number 5, 7 and 9 and writing here separately, and I can leave all the uninformative side behind. Now I will focus only on these columns 5, 7 and 9. Now let's see how I can conclude that which type of relationship 1, 2, 3 and 4 are sharing. So for that one, what we can do, we can generate this type of the hypothetical tree. Hypothetical tree means what are the relationships should be there. What kind of the relationship 1, 2, 3 and 4 may share. Maybe 1 and 2 ancestors is common. Like you can guess all the type of the possible uh, relationship, means 1 and 2 having the common ancestor, 3 and 4 having the common ancestor, or 1 and 3, 2 and 4 have the common ancestor, 1 and 4 have the common ancestor, 2 and 3 have the common ancestor. So these are the only three possible ways. These are the three possible relationship in the all four sequences. If you write down like 1 and 4, or if you shuffle up, then you can try by your pen and paper that there only will be the equivalent trees. So these are the only three possible relationship between the sequence number 1 to 4. Apart from that, none of the other relationship is possible. But so here is three options. Now I don't know actually which type of relationship all the sequences are sharing. So what we can do? Once I have written 1, 2, 3 and 4. So here if you see the sequence number 1, you have three bases G, G and A. So here I have written G, G and A. In the column, in the 2, sequence number 2, here I have written G, 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 G. At the position number 3, in the sequence number 3, you can see here thus A, C, A. So here I have written A, C, A. And in the 4, here it is A, C, G. So here I have written the A, C, G. Likewise, in the 1, you can put AGA, you can put ACA, you can put the value of sequence 2 is triple G, or 4 it is ACG. So likewise you write down all the things. Now the point number 1, now what we can do, I want to check whether this is the true relationship between the 1 and 2 and 3 and 4. So first of all, you compare between these triple G and GGA. So if you see you always make practice while you are trying to solve the this kind of the algorithm. You always try to move from bottom to top. You compare the bottom sequence with the top sequence, and this is the common ancestor of taxa 1 and taxa 2, or sequence 1 and 2. So here I can see in the triple G and GGA how many mutation is there. So here you can see GG is conjugate, but on the place of G there is the one mutation, that rather than G here it is A, means there is how many mutation? There is one mutation. This algorithm is based on one hypothesis that is known as molecular clock hypothesis. What molecular clock hypothesis is? it says that the pace, the uh, rate of mutation is constant. So what we do, if we consider that the ancestor of 1 and 2 is common, so this is the common ancestor of 1 and 2, and during the evolution there was one mutation in the sequence 1, right? So what I can do with this, if this is the ancestor, so there is no mutation in the 2, but there is one mutation in the sequence number 1. So what I can do here, I can write down that with this parent ancestor there is no mutation, and so it is triple G, and with in the sequence number 1 there was one mutation. The same way I can compare between the ACG and ACA. Here you can see ACG and ACA there is one mutation, so there is no mutation in the ancestor and the sequence number 4. But here if you compare with ACG with ACA, there is one mutation. So now you can count that how many total. And one more thing, now you compare with this common ancestor and this common ancestor, ACG, ACG and triple G. How many mutation you can notice? You can notice two mutation, right? Here you can notice two mutation. So here is the two mutation. Now you can count that how many total number of mutations are there in this particular relationship. So here I found 1 and 2, 3, 3 and 1, 4. So total in the tree 1, I got total 4 mutation. All right. Now, suppose now I want to check in this the relationship number 2. Here you can see that ACA and GGA. How many mutation are there? You can check it out. Rather, on the place of A, here is the G. On the place of C, here is the G. So how many mutation? And this A is conjugate. So there is two. How many mutation? Two mutation. So once you have the two mutation, what you can do? You can divide in two equal parts, 1 and 1. So ACA, ultimately what I told you, that you always move from down to means bottom to top. So you have to convert this ACA into GGA. So if you want to convert ACA to GGA, one mutation you can put. So A can convert it into G, and this common ancestor will be GCA. And once now you have to convert GCA to GGA. So again you can change this C and you will get sequence number 1. So 1 and 1. Likewise, if you say in the ACG and triple G, the same way, how many mutations are there? Two mutation. So you can divide in two equal part, 1 and 1. All right. Now you can compare between GCA and GCG. So GCA and GCG, there is one mutation. So how many total number of mutation is there in this particular relationship? There is 5 number of mutations. Now see in the relationship number 3rd. Here you say GGA and ACG. Here you see, very interesting. ACG and GGA, here you find total 3 mutations in the 1 and 4. Now, previously I told you in this case, if there is 2 mutation, I can divide into equal part 1 and 1. In this condition, there was only one mutation, so I can categorize into 0 and 1. So what you do, once there is the mutation possible in the odd number, if it is in even number, then no problem. You can divide into equal part. If it is in odd number, then the bottom number you can give them less number, and in the top one you can give them more number. Like if 3 mutations are required, then you can put 1 and 2. If it was suppose 5, for example, then it should be 2, that downside should be 2 and upper side should be 3. Suppose it was 11, then what should be the combination? It should be 5 and 6. If it was 13, then it would be 6 and 7. Alright. So likewise, if I want to convert ACG to GGA, then what I can do? First, start mutating from here. So A you can convert into G. So now it is G. I think here I have committed one mistake. Here should be GCG. Let me make the changes so that will not get the confusion. So it should be the GCG. All right, I think it is correct now. So now it is, yeah. So now it is GCG. Now you can compare between the GCG and GGA. So now this C should be converted into G, and this G is converted into A. All right. So it is now the same way ACA and triple G. How many number of mutation is required? 3. So 1 and 2 combination should be there. So if you create one mutation, so A can be converted to G. So here what I do, I think here is the also one problem. So what I can do, let me make the changes. I think here is again more confusion. So here it should be GCA. Okay. So now it is GCA. Only one mutation I take. I put it one mutation. So ACA is converted into GCA. Now you want to convert GCA into triple G. Then how many number of mutations should be there? 2. And now you compare between the that how many mutation is how many mutation are there in the ancestor of 1 and 4 and in the 2 and 3. So GCG and GCA, here you can put, see how many mutation, only one mutation. So now you can count total number of mutations in 7. Now you compare that which relationship having the minimum number of mutations. So the minimum number of mutations is having the tree 1, means you can conclude, means to establish the evolutionary relationship among the sequences, the minimum number of mutations required, that will be the maximum parsimonious tree, means here you can conclude that tree number 1 is the right answer, means 1 and 2 having the common ancestor and 3 and 4 having the common ancestor. So the correct relationship between the 1, 2, 3, 4 is this one. So that's how you can conclude this is the evolutionary relationship among the 4 sequences. And this one is not correct. This one is going to reject. So this is the true evolutionary relationship by using the maximum parsimonious tree. So you can now interpret the maximum parsimonious tree, since the minimum number of mutation was required in the tree number 1. So that was the correct answer. So I hope the maximum parsimony algorithm is clear to you. You try to search the books or over the internet, you will find a lot of examples of the maximum parsimonious tree. Try to solve by your own that which type of relationship is there in the sequences. So we'll see you very soon in the next algorithm to solve the maximum parsimonious, to solve the phylogenetic analysis related problems. So see you soon. Take care. Namaskar. Jai Hind.